All right, welcome to the recording for chapter 27. We are going to be discussing the male reproductive system today. And there's only one recording for this chapter. We keep it kind of short and sweet. So we're going to talk about sexual reproduction and development, kind of a general overview. Might remember this from Intro to Biology, hopefully. So when we're looking at the males and the females, we have what's called gametes, which are your sex cells. These gametes are going to combine their genes to form what's called a zygote. One of the gametes is motile, so generally the sperm for humans is going to be the motile gametes. The other gametes is going to contain nutrients. So in the development of the egg, we actually see one dominant egg or ovum that's developed, and it's large and it contains a lot of cytoplasm. So it has lots of the organelles and the nutrients that's going to need to sustain this fertilized zygote. So, and then the sperm being motile, it has a flagellum to help propel it and move it to get it towards the ovum for fertilization. And we call that the spermatozoon. Okay, so when we're looking at males and females, we have what's called our primary sex organs. These are going to be your gonads, so the testes or the ovaries. And then the secondary sex organs, they're going to be also necessary for reproduction, but they don't produce these the gametes. So, but they help. So for males, it's ducts and glands that are going to deliver the sperm. And then for females, it's uterine tubes, the uterus and the vagina. So the uterine tubes are important for the released egg from the ovaries, and the uterus is important for fertilization and for housing a developing fetus, and of course the vagina is important for sexual intercourse and receiving the semen. So with our secondary sex characteristics, excuse me, now this is not the sex organs, these are the sexual characteristics. Some of the changes that we see that are distinguishing for the sexes. We see in both sexes we have pubic and axillary hair, and also the associated scent glands that go along with this. We can see changes in the pitch of the voice. And with these pubic and axillary hairs, we have these scent glands that also release pheromones, and these pheromones cling to the hair of the pubic and axillary regions, and so your pubic hairs and armpit hairs, and then it keeps those pheromones more closely associated with the body. For males, we tend to see more facial hair, coarse, visible hair on the torso and the limbs, and kind of more muscular physique. For females, it's more of a distribution of body fat and breast enlargement. So those are some of the secondary sex characteristics that we tend to see. Also really playing an important role in mate attraction. So for the males, now we kind of see some of the general features of reproductive systems in general. The male reproductive anatomy, we're going to spend just a little bit of time on this. I know you get this in lab as well. So the testes are located they're housed within what's called the scrotum. And the scrotum has some muscle that we're going to be discussing that can either draw the testes upward or let them hang a little bit lower. That's important because for sperm production, the temperature needs to be slightly lower than the body temperature. So depending upon the body temperature, it allows, and the external temperature, it allows to maintain the right temperature for sperm production. This is showing us our system of tubes. This is the vas deferens or ductus deferens that then leads into and kind of goes alongside the bladder. And then, so this is the pathway the semen is going to take, the sperm, I should say. Then it's going to join with the seminal vesicles. About 60% of the fluid that's going to be ejaculated comes from the seminal vesicles. Then it enters into what we call the ejaculatory duct, which goes through the prostate. And at this point, and then also the prostate gland is going to also inject some secretions as well. And also we have the bulbourethral gland. So now we follow this pathway. This is the urethra. that then follows the pathway on out. So we'll go in more detail about this, but this is kind of the general setup and the general features of the male reproductive tract. So we've talked about the scrotum containing the testes, and all of this external genitalia occupies the perineum, kind of a diamond-shaped area between the thighs. And we also see what's called the spermatic cord, which has a lot of important features to it. It has a lot of the ductus deferens, blood vessels, lymphatic vessels, and nerves as well. So this shows us the perineum, this little diamond-shaped structure between the thighs. And if we're looking at the scrotum, this is showing us the spermatic cord, and we have this important muscle, which is called the cremaster muscle, located right here. And also, more deeply, we have the vas deferens, or the ductus deferens, right here. And then we have blood vessels and nerves that make up the spermatic cord as well. So when we take a closer look at the cremaster muscle, this cremaster muscle is what's responsible for drawing the testes upward or suspending them downward, depending upon the temperature, because, like we said, the sperm need to be at a slightly cooler temperatures, about 35 degrees Celsius instead of 37 degrees Celsius. And you can also see the cremaster muscle here as well. And then more superficially to that, we have what's called the dartus muscle. And this dartus muscle is a little bit deep to the scrotal skin, and this makes it more wrinkled on the scrotum. And it also contracts when it's cold, and so forth. So it can also provide with some insulation and so forth. Okay, so when we're looking specifically at the testes, we have lots of different types of cells. It is both, this is kind of crazy, it's both an endocrine and an exocrine gland. So it's endocrine because it releases hormones like testosterone. And it is an exocrine gland because it actually releases, it releases an entire cell, which is the sperm. Okay, so once again, produces sex hormones and produces the sperm. And it's also, we call this type of gland a cytogenic. So cyto means cell, genic means creating it. So it creates a whole cell and then excretes it. It's an exocrine gland. So we also have what makes up the testes, seminiferous tubules. And these seminiferous tubules is where we're going to see the production of the sperm. You have 1 to 3 of them in each of the lobules. You also see microscopically what's called interstitial cells or Leydig cells. Your interstitial cells produce testosterone. Now, we have another type of cell called the sustentacular or Sertoli cells. And I call these, I think your book may as well, the nurse cells. These are the nurse cells. So kind of makes sense. They protect the germ cells, and they also will promote their development. So how these nurse cells protect the sperm is they form something called the blood-testes barrier. And what the blood-testes barrier does is it prevents antibodies from getting to the sperm. And the reason for this is that the sperm become genetically unique. Once we see that they're haploid cells, they're now genetically unique from the body cells, and we do not want antibodies manufactured because that could actually lead to someone becoming sterile. So we have to protect these unique cells from the body's own immune system. Okay, we're going to talk more about that as we go. The reedy testes, they are a network embedded, and this is going to collect the sperms from the seminiferous tubules. So this is just showing us the testes and along with its other structures. So here we can see the testes, this is the scrotum, and this structure is showing the epididymis. This is going to contain developing sperm after it is released from the testes. They develop within here for several days. They migrate from what we call the head of the epididymis down the body into what we call the tail of the epididymis. From there, they then enter into the ductus deferens or vas deferens. So if we look closely at the histology of the testes, we can see here, this is showing us one seminiferous tubule right here. So this is where we have development of sperm. And we can also see some of those cells that are going to help the development and protect the sperm as well. So the sustentacular cells are right here and right here. You can see those. And then the interstitial cells are located right here. So here's the interstitial cells. And we will talk more about those interstitial cells here in a second. So to give you a little bit of preview of what these two different types of cells do, your interstitial cells are going to produce lots of testosterone. Now, this testosterone encourages the development of the sperm, but the thing is, you have to have a binding protein that binds testosterone and keeps it in the seminiferous tubules to stimulate that production of sperm. So, like we said, the interstitial cells, these guys are going to manufacture testosterone, but then these sustentacular cells, and we can see more of them here and here and here, they are going to produce something that's called an androgen binding protein, ABP. So they produce an androgen binding protein, and what that does is it makes the testosterone that's manufactured by these cells, and it makes it stay, it binds it and makes it stay in these seminiferous tubules so that they can encourage the development of the sperm. And we can see some of those developing sperm. Here's spermatids that are right here. Okay, and what happens is the sperm develop from this outer wall and migrate inwards and complete their, well, and they're really not done with their development. They actually continue it once they're released, but their maturation and everything process does occur, and it works its way from outward to inward. And then this is the lumen where the cells kind of let go and then follow the system of tubes. Okay. So when we're looking at this network, this is showing us a lobule of the testes, and all of this kind of looks like macaroni. Those are the seminiferous tubules. This is where we see the sperm production. So in this, those seminiferous tubules, we are seeing a cross section of them in this image. So we've got 1 to 3 of the seminiferous tubules that are inside of each lobule. Now, once the sperm are produced, they're going to collect into this system that's called the reedy testes. Okay, so that is right here. Then the sperm enters into the efferent ductules. Now, we've learned efferent means leading out of, so it makes sense it's leading out of the testes. Then it enters into the epididymis. So it goes into the head of the epididymis, then it goes into the body of the epididymis, and then down into the tail of the epididymis. And then it follows this pathway, like we've shown before, through the vas deferens or ductus deferens. Okay. So when we're so to talk a little bit more about this epididymis, it can take about 20 days for the sperm to reach the tail of the epididymis, and it's actually about 18 feet long. So this epididymis has lots of surface inside of it. Again, so the sperm is really continuing its development throughout this whole process. So it's matured and it's stored here. Now it can stay fertile for about 40 to 60 days. If there is no ejaculation, then the sperm that are within the epididymis, they kind of disintegrate, and the body just reabsorbs the sperm that were not ejaculated. Okay. So that's kind of the pathway that it leads through when we're looking at the testes. Okay. So we then see from our vas deferens, we're then going to enter more of the body cavity. We go alongside the bladder, and this is where we're going to meet up with the seminal vesicle, and we already kind of mentioned this, the prostate gland and the bulbourethral gland. And we'll talk about the importance of those and what they contribute to it here in a second. So this shows us a much better view. We can see that it kind of, this is a posterior view, so this is going alongside the back, the posterior side of the bladder. And when we're looking at these accessory glands and how they contribute, if you're looking at the seminal vesicles, we mentioned this is already about 60% of the semen is made up from fluid ejaculated or added to it from the seminal vesicles. And it's more of a kind of a yellowish fluid secretion. And something to add to that, the seminal vesicles are going to have lots of sugars, so fructose and other sugars. This is going to be important for the mitochondria of the sperm. Once the sperm is inside the female, then that sperm is activated and it's going to need energy. So it gets this fructose from the seminal vesicles that's along with it, so it can have lots of energy for movement. When we are, excuse me, talking about the prostate gland, so the prostate will contribute about 30% of the semen, and it's a very milky secretion. And so when you're looking at what the prostate helps with, it will help buffer against the acidity in the vagina. The vagina is slightly acidic, so it can bring it from a pH of 3.5 to a pH of 7.5. So it buffers it quite a bit. The importance of this is that the sperm are activated by a higher pH. So to activate them, this prostate helps buffer that acidity so that the sperm become activated. So we also see the prostate will contribute a couple other things. It will contribute a clotting enzyme. Now this clotting enzyme is important because it helps the semen to kind of adhere and stick. It becomes sticky and it adheres to the vaginal wall. But then it also produces something called serine protease, which will liquefy and break down this clots, the clotting enzymes and everything that's clotted within 30 minutes. So now it's liquefied and then the sperm can kind of be on their way. So the last one that we're going to look at, this is the bulbourethral gland, which is right here. Now what this helps with is because the urethra is a shared tube, it's shared by both the reproductive organs and also the urinary. We've got urine that comes through this same opening. So what the bulbourethral gland will do is it will buffer if there's any acidic urine that's in the urethra, the spongy urethra, it'll buffer that pH so that the sperm are still viable. It also provides kind of a slippery fluid for lubrication during intercourse. So that's all coming from the bulbourethral gland. So that really is what makes up the semen. It's sperm, but it's also a lot of these fluids. Again, 60% seminal vesicles, 30% coming from the prostate. The other 10%, you're going to have bulbourethral secretions and also the sperm itself. Okay. So the next thing we're going to talk about is the penis and the structures of it. So, and real quick, let's talk about the prostate again. So a lot of, when we're talking about the prostate, we can see benign, like a non-cancerous enlargement. By the age of 70, about 90% of males can have a non-cancerous enlargement. Problem with this, they call it benign prostatic hyperplasia, BPH. And this enlargement of the prostate can compress on the urethra. So it can compress on this urethra, and it also can obstruct the urine flow, and then there can be an increased chance of bladder and kidney infections and so forth. So lots of complications that can come about from that. When it comes to cancer, if it's caught early, there's a good survival rate. They check for prostatic enzymes that are in the blood when they're doing tests and looking for prostate cancer. Okay, so when we're looking at the penis, there are some major components to it. So we have 3 kind of cylinders that are filled with blood vessels. And this is the erectile tissue. It fills with blood when someone's sexually aroused, and this is what causes the erection. And we have one corpus spongiosum, which is located right here. In the center of it, the urethra runs through it. Then we have on either side, we also can see what's called the corpus cavernosum, and there's a deeper artery that is found within there. So these are filled with the erectile tissues. Now, if we're looking at the external portion of the penis, then we can also see that there is kind of the glands of the penis, which is right here, and the external urethral orifice, and then we have what's called the prepuce. And this is a little bit acute extra skin. So when somebody is circumcised, they will remove this skin folding. And so that kind of is loosely attached, allows for expansion of the penis. All right, so now we're going to get into the endocrinology of the male reproductive tract. So these are going to be some hormones that hopefully refresh your memory from Anatomy and Physiology 1. We've got a couple of hormones, 3 of them that are dealing with the hypothalamus and your pituitary. So the major one that we're looking at is called gonadotropin-releasing hormone, or GnRH. And this hormone, gonadotropin-releasing hormone, it originates from your hypothalamus. So we're going to take a look at this picture here. So we're going to look at the green arrows first. We're just going to talk about those for a second. So this is showing us the hypothalamus, and so we have it kind of right in here. And gonadotropic releasing hormone is the hormone that's produced by the hypothalamus. It's going to flow through a network of blood capillaries called your hypophyseal portal system, and it flows down into the anterior pituitary. Now what it triggers is it triggers then the release, stimulates the release of 2 hormones. One is called FSH and one is called LH, and so they're produced and released by the anterior pituitary. So we're going to first take a look at your luteinizing hormone. So LH stands for your luteinizing hormone. So this hormone comes down, flows through the bloodstream, and it's going to bind to receptors that are on the surface of your interstitial cells. So remember, these cells were in between the in between where the sperm were being produced. So the interstitial cells, if you remember, they manufacture testosterone. But we also have to have a way to keep that testosterone locally. Some of it's going to flow through the bloodstream and go throughout it. But we also want to keep some of it locally in those seminiferous tubules. That was a word I couldn't think of. So this is where the sustentacular cells come in play. The sustentacular cells were inside the seminiferous tubules, and they release, here's that little enzyme, that little androgen binding protein that is going to bind some of this testosterone and keep it locally in. And that is going to then stimulate sperm production. So we're going to get spermatogenesis as a result. We're going to get the sperm production. Now, what also happens? The sperm is in the bloodstream. So this will stimulate the libido. It also helps with the development of your secondary sex organs and characteristics. But it also has a feedback mechanism. So testosterone will have a negative feedback mechanism, and what that means is now we've created the testosterone, and now we can say, okay, the hypothalamus can detect and say, guess what? We've done our goal. We wanted to have testosterone, and now we have it. And so this is a negative feedback mechanism to reduce gonadotropic releasing hormone secretion, and also the pituitary being sensitive to it. So we also see, looking at the red arrows, I'm going to get rid of this now. Looking at the red arrows, the sustentacular cells also release a hormone that's called inhibin. it inhibits. So inhibin will selectively come up here, and it will stop the secretion of follicle-stimulating hormone. So selectively, it only stops the follicle-stimulating hormone. The importance of this is that if we've got good sperm production and we don't need more sperm production, we can stop follicle-stimulating hormone, so then the sustentacular cells reduce the production of androgen-binding protein. There's less testosterone that's kept around the seminal vesicles, and so we can lower the sperm production and say, okay, we've got enough. And it does this inhibin does it without affecting luteinizing hormone. And the importance of this is that we can still keep testosterone levels elevated. So we can only affect the sperm production and not affect the testosterone levels, because that's an important aspect of it. So I hope that makes kind of sense with the negative feedback mechanism of testosterone, but then also the inhibin affecting the secretion of follicle-stimulating hormone. Okay, so when does all this happen? Around when we say kind of puberty. Puberty is generally around the ages of 10 to 12 in boys. We see a surge in pituitary hormones, these follicle-stimulating hormone, luteinizing hormone, and it kind of really awakens the reproductive system. Now, so once we start seeing follicle-stimulating hormone and luteinizing hormone being produced, we're starting to see the sperm production, we're starting to see testosterone production, and then generally we say that this is, so we see kind of around the age 13 that there's enlargement of the secondary sexual organs. And then around the age 14, and none of this is set in stone, but around the age of 14, we start to see the first ejaculation. And so between that time, again, 10 to 12, we start to see pituitary hormone secretion. Then we get enlargement of the sex organs, and then first ejaculation. That's the timing of puberty. Now, the bigger window is what we call adolescence, and that's when we first see gonadotropin being released. So this whole process that's beginning, all the way until a person reaches their full height. That is adolescence. It's kind of a bigger picture. Now, once we get into adulthood, this testosterone is really going to sustain your reproductive tract, and sperm production and libido, kind of as we've shown in this hormonal relationships graph. And we do see after the age of 20, there's a decrease in testosterone production, there's a decrease in cells, there's a decrease in sperm that corresponds with this. And it has been documented, and some authorities believe that some men go through what's called andropause in the early 50s, which is a reduction in these hormones and a reduction in the sperm count as well. Not to say they could not still get someone pregnant, it can happen, but their sperm count does tend to drop with the testosterone levels as they drop. Okay, so what this takes us into next is spermatogenesis, and this is the production of sperm and semen. And there's really kind of three principal events that are going to happen. We're going to take these larger germ cells, and we're going to make them very small and mobile with the flagellum. We're going to reduce their number of chromosomes they're going to be haploid, which means they have one version of each of the 23 types of chromosomes instead of two. And we're going to shuffle the genes and make them unique. So for every one germ cell, we can end up getting 4 sperm cells produced. And this is going to take you back to intro to biology. We're not going to spend too much time on this because you all are capable of reviewing this. You hopefully have this in intro to biology. This is showing us meiosis 1. This is the first division. If we're looking at a germ cell, it takes us through the process of ending up with 2 cells dividing from it. And what we are seeing here is we're only showing 2 homologous chromosomes. Keep in mind there's 23 pairs. We're only showing 2 here. And so they join up with their homologous pairs. And we can see at this point some crossing over that happens. So you can see how this is red here and this is blue. So that creates genetic diversity. and then they line up in what's called the metaphysis and the metaphase, the middle portion of the cell. And then we can see the homologous pairs have separated. And then the cytoplasm separates. We call this cytokinesis. Then both of these cells divide as well. So if we're walking these through it, we have these, the cells, once again, the chromosomes line up along the center line. This is metaphysis 2, the equatorial plate. Then we have division again where they separate. Okay, we can see that here, they're pulled apart. And now we've got cytokinesis happening. This is telophase 2. And now, if you look at each one of these, we have 4 cells, and look at their chromosomes. They have one version of each type of chromosome. So that's kind of the importance of them. So we can have one germ cell give us 4 sperm. And I document all this. I'm not going to go through all this because this is intro to biology. But what I am going to talk about is how we are relating this to what we call spermatogenesis. So what happens here, this is showing us the outer wall of the seminiferous tubule, and we have located right here spermatogonium. Okay, and these guys are located against, really against the wall. So what's going to happen is we are going to see the spermatogonia divide, and it does so by mitosis. This is not by meiosis yet. This is just normal cell division where they're going to have the same genetics. But once we have this division, we call one the type A spermatogonium and one the type B spermatogonium. Type A is going to stay back. It's going to stay located against the wall. Type B is going to migrate more inward. And this type B spermatogonium is going to be destined to divide. Okay, so this type B spermatogonium is going to undergo meiosis 1, and that's what we're seeing here, meiosis 1. And notice how it says 2N. That means that it has 2 versions of all the 23 types of chromosomes. But now, once we have this first division, it's 1N, because now they only have one version of all 23 types. Now that version is replicated, and we can see that here, going back to this picture. We can see, let me erase the ink on this slide. So we can see that these chromosomes are replicated. So this is one, we call it a sister chromatid. This is one sister chromatid, this is the other. So they're replicated. But now this cell only has one version, and this one has the other, even though they're replicated. It's the version of the chromosome. So now we call them N, okay, because they only have one version. They only have blue or red, they don't have the other one. Now, they do have crossing over, but then they're still missing that other portion of it. So, now we call these secondary spermatocytes, okay? Once they're getting ready to divide, we call them primary spermatocytes, and now we call them secondary spermatocytes once they have this first division have undergone it. So then those are going to undergo meiosis 2 and give us what we call spermatids, and there's 4 of them altogether. So now these 4 are going to migrate inward, and they're going to then be called sperm, and they're going to undergo a process called spermiogenesis, in which they grow a tail. They also use a great deal of their cytoplasm so that they're very lightweight, and they're motile, and they have that flagellum, and they've got lots of mitochondria to generate energy. Now, the other thing I want to mention throughout this process is something called the blood-testes barrier. So the blood-testes barrier, we can actually see this here. Now, all of these cells, and let me get rid of this ink on this slide again. If you look at this whole entire thing here, this is one this is showing a sustentacular cell. Here's another sustentacular cell. Okay? So what's important about these guys is they have tight junctions in between them. So that's what this represents. These little tight junctions are forming the blood-testes barrier. This is the barrier between the bloodstream and really, I mean, having contact with the sperm, because these are now becoming genetically unique cells that we don't want antibodies formed for, and we don't want the immune system to mount an attack against it. So that is an important aspect of it. Now, this whole process, going from this to this, takes about 70 days to produce these sperm. Now, you also have, if you kind of talk about the numbers, a normal sperm count is 50 to 120 million sperm per milliliter. If someone has 20 to 25 million sperm per milliliter, that's considered to be infertile. So, great deal of numbers. Okay, so what happens in spermatogenesis? So we just talked about spermiogenesis, the production of the sperm. Now we're looking at spermatogenesis, which is the changes that happen to these sperm. So going from what we call spermatids to spermatozoa. So we've mentioned already that we're going to discard, we're going to shed some of this excess cytoplasm because we want the cell to be very lightweight, and also think that the egg is contributing a great deal of the cytoplasm. So the sperm doesn't need to, it's not contributing it. We're getting a great deal of it from, really all of it from the female egg. We're also going to see the development of something called an acrosome. This acrosome on the head of the sperm is important because it consists of lysosomes that are going to, once we have fertilization with an egg, it's going to break down the cell membrane, and that way we can get the nucleus. So here's the nucleus. We want the nucleus to get inside of the egg for fertilization, and the acrosome is important for kind of eroding away the cell membrane and really getting that that nucleus inside of it. So we also see gobs of mitochondria, and then we also have, this is showing the mature sperm and showing the tail. So those are some of the key features of the spermatozoan. So, and when we look at the tail, we can see portions of this. This is the principal piece of the tail, and this is the end of the tail, and this region is called the mid piece of the tail. And we can see this in a real sperm, which is really cool. There's the acrosome, there's the nucleus, lots of mitochondria, gobs and gobs of mitochondria. Okay, and we kind of already went through the semen and what it's made up of. It's 2 to 5 milliliters expelled during ejaculation. And what makes up it, here's kind of that sperm count that we mentioned. The prostate buffers the acidity to activate the sperm. It's a milky fluid. The seminal vesicles are going to have more of that viscous yellow fluid. Lots of fructose and sugars that are there. And also, we've got the seminal vesicles are going to have something called proseminogelin, and this is what really makes it sticky. And the prostate has the clotting factor that kind of activates it and makes it sticky. And then the prostate also has something called serine protease that breaks down and liquefies the stickiness 30 minutes after ejaculation so that the sperm can be on the way. So the sperm aren't actually motile until they're ejaculated. Once we've got a buffer in the pH, then that kind of activates them, and then they start swimming, and we also have the fructose and sugars to power their mitochondria. So that is the basics of what we're going through this chapter. I know your textbook goes through a lot more, but given our short timeline, that's what we're going to limit it to. All right, have a good